Good morning, students. Myself, K.K. Lekha, Assistant Professor in the Department of Biomedical Engineering in Erode Engineering College. In today's class, we are going to see about the microbiology and structure of bacteria. We are going to see about the structure and components of bacteria and also functions of bacteria in this class. Components of bacteria like cell wall, cytoplasms, ribosomes, flagellas, etc. we are going to see about in this class. Before that, we have to know about the microbiology. Microbiology is the study of microscopic organisms such as bacteria, viruses, archaea, fungi, and protozoas. Basically, there are many types of microbiology branches like bacteriology, immunology, mycology, nematology, and pycology, etc. In bacteria, it is the study of bacteria. Bacteriology, it is the study of bacteria. In immunology, the study of immune systems. In mycology means it is the study of fungi such as yeast, molds, etc. In nematology, the study of nematodes like roundworms that are comes under the nematology. In phycology, the study of algae. Depending upon the cell life and functions, characteristics of cell, we are divided the cell into two types. Basically, all living things that are single or multicellular cells are made of cells that share some common characteristics like basic shapes, internal contents, DNAs, etc. Basic shapes like spherical, cubical, cylinders, and also the internal contents are cytoplasm surrounded by a membrane like that. And DNA, it has chromosomes, ribosomes, metabolic capabilities. Depending upon these characteristics and also the lifetime, it is divided into two types that are eukaryotic and prokaryotic cells. In eukaryotic cells, these are cells that possess the true nucleus with the membrane bound organelles. Eukaryotics are either be unicellular or multicellular. But in prokaryotic, it is characterized by the absence of nuclear and do not possess membrane bound cellular organelles. Prokaryotes are exclusively unicellular, which means in prokaryotics it has no nucleus and other membrane bound organelles. But in eukaryotics, it has a nucleus cells and also membrane bound organelles. The examples of prokaryotic cells are bacteria and archaea. In eukaryotics, the examples are protists, fungi, plants, and animals. Everything except prokaryotic cells comes under eukaryotic cells. Okay, let's go get into the bacteria. Bacteria are the microscopic single cellular and unicellular prokaryotic organisms that exist in their millions in every environment, both inside and outside other organs not visible with the naked eye. They are divided by binary fissions. Some bacteria are harmful, but some are useful for useful purposes. They support many forms of life, both plant and animals, and they are used in industrial and medical processes. There are four common types of bacteria are Coccus, Bacillus, Spirilla, Vibrio, and other common types of bacteria. In Coccus, there are spherical bacteria, spherical structured bacteria. The Bacillus are the rod-shaped bacteria. In Spirilla, it is the spiral-shaped bacteria. In Vibrio, the comma-shaped bacteria. It is a comma-shaped bacteria. Okay. The figure shows the structure of bacteria. In structure of bacteria, it contains inside and outside the cells. In inside the cells, it has a nucleoid, DNA coiled nucleoid, ribosomes, and cytoplasms, and plasma membranes, and also cytoplasmic inclusions. And in the outside the cell wall, it has a capsule, cell wall in red color, it shows in red color, and flagellums are long hair like structure, and villi, basal bodies, etc. Okay, it is divided into outside cell wall and inside cell wall. Okay, outside cell wall, it has capsule, flagella, villi, sheath, prostag, stacks. In inside cell wall, it has a cell wall, cytoplasmic membrane, nucleoid, mesosomes, ribosomes, cytoplasms, and spores. In capsule, it is a zero point 0.2 micrometer thickness viscous layer, outer layer to the cell wall. It is outer layer to the bacterial cell wall. The capsule is 98% water and 2% polysaccharide or glycoprotein or polypeptide, or it has both. There are two types of capsules, that are macros capsules and micros capsules. In macros capsules, it is a 0.2 micrometer thickness or more. It is visible under the light microscope. In micros capsule, the thickness is less than 0.2 micrometer. It is visible only under the electron microscope. The functions of capsules is it helps in attachments as well as prevent the cell from desiccation and drying. Next is flagella. It is 15 to 20 nanometer hair-like helical structures emerges from cell wall. Flagella is not straight but is helical. It is composed of flagellin protein, like known as globular protein. Flagella is has three parts: basal body, hook, and filament. The functions of flagella is it helps motility of the bacteria. There are so many types of bacteria depend on the flagella. It is shown in the figure. First one is Atrios. Atrios has bacteria without flagella. Example, Xylella is example for Atricus. In Monotrichus, the name itself, it has a single flagellum. Example is Vibrio chlorale. In Amphitrichus, it has single flagellum on both sides. Examples Algalisans and Facules. Lophotrichus, it is a tuft of flagella at one or both sides. Examples Spirillum. And the last one is Petricus. Petricus is numerous flagella all over the bacterial body. If we have the flagella, numerous number of flagella and all over the bacterial wall is known as Petricus. Okay, next is pili or fimbriae. Pili are hollow, filamentous and non-helical structure. They are numerous and shorter than flagella. Pili is the characteristic feature of gram-negative bacteria. Villi is composed of filin protein. The functions of villi is attachment. In attachment, villi helps the bacteria to attach the host cell surfaces and also possess antigenic properties. And is prosthegae. Prosthegae are semi-rigid extension of cell wall and cell membrane. One bacteria may contain one or many prosthegae. The functions of prosthegae are increase surface area of nutrition absorptions, and also it helps in adhesions asexual reproductions. And the next bacterial component is stack. It is a non-living ribbon-like tubular structures. It is formed by excretory product of bacteria. The function of stack is it helps in attachment of solid surfaces. And the next one is cell wall. It is an important structure for bacteria because it products the entire components in the inside the cell wall. Okay, it gives shape to the organism and also it gives shapes to the organism. On the basis of cell wall composition, bacteria are classified into two groups. There are gram-negative and gram-positive bacteria. In gram-negative bacteria and gram-positive bacteria, the differentiation is shown in the slide. The gram-positive bacterial cell wall, it is a composition of positive bacteria like peptidoglycan and lipid teichoic acid. In gram-negative cell wall, the composition of negative bacteria, that is peptidoglycans, outer membrane is lipid protein lipopolysaccharide. The examples of gram-positive bacteria is Escherichia coli, Vibrio cholerae, Salmonella and Pseudomonas. The examples of gram-negative bacteria is Staphylococcus aureus, Mycobacterium tuberculosis, Streptococcus pneumonia. Okay, next is cell membrane. Cell membrane is the inner thin layer Cell membrane is inner thin layer that lies inside the cell wall and encloses the cytoplasm. It is known as cytoplasmic membrane or plasma membrane. It has a thickness of 18 nanometer and the cell membrane of bacteria is composed of phospholipids and proteins. The functions of cell membrane is the selectively permeable as it allows to pass selective substances. Next is nucleoid. Nucleus is the most important part of the cell. It controls and directs all the cellular activities and stores hereditary information of the cell. Bacterial nucleus is known as nucleoid. It lacks nuclear membranes, nucleoplasms and nucleus. Bacterial DNA is lacked histone protein. The functions of nucleoid is it contains and stores hereditary information of the cell and it controls the cell activities. Next is ribosomes. The bacterial ribosomes is of 70 S types. Ribosomes are rounded granules found freely floating in the phytoplasms. Ribosomes are known as universal cell organelles because it is found in the both bacterial cell and eukaryotic cells. The functions of ribosomes are it helps in the protein synthesis. The next one is mesosomes. Mesosome is a spherical or round sac-like structure found commonly in the gram-positive bacteria. The function of the mesosome is it is the site for respiration in the bacteria cell. The next is cytoplasm. It is the colorless viscous fluid present inside the cell membrane. All the cell organelles and inclusions are found floating in cytoplasmic fluids. It contains proteins, lipids, minerals, nucleic acids, glycogens, waters, etc. The functions of cytoplasm is it helps distribute water, oxygen as other substances through the cells. Next is spores or endospores. Spore is a metabolically dormant structure produced during unfavorable condition by the process called sporulation. Sporulation occurs during late log phase or early stationary phase. Under favorable conditions, spore germinate to give vegetative cell. Okay, next is we are going to see about the difference between the gram positive and gram negative bacteria. In gram positive bacteria, it is a simple cell wall. But in gram negative bacteria, it has a more complex cell wall. In gram positive bacteria, it is a thick peptidoglycogen cell wall layer. But in gram negative bacteria, it is a thin peptidoglycogen cell wall. In gram positive bacteria, it has no outer lipopolysaccharide cell wall layer. But in gram negative bacteria, it has an outer lipopolysaccharide wall layer. In gram positive bacteria, it retains the crystal violet or iodine. But in gram negative bacteria, it retains safranine. Gram positive bacteria appear like blue or purple in color. But in gram negative bacteria, it is appear like pink or red in color. The classification of bacteria according to its shapes is shown in the slide. In the spear-like structure, it has a diplococci or streptococci. Diplococci means the only two round blasts, etc. And also it has a tetrad, sarkia, and staphylococci. In rod-like structures in bacterial cell, it has a chain of bacilli, Bacillus anthracis, and flagella rods, and spore former. In spiral-like structures, it has vibros and spirilla and spirocactus. In cocci, these types of bacteria are unicellular and also spherical or elliptical in shape. It has many types. First one is monococcus. There are also called micrococcus and represented by single discrete round shape. Examples is Micrococcus flavus. In Diplococcus, the cell of Diplococcus divides once in particular plane, and after division the cell remain attached to the each other. Example is Diplococcus pneumonia. In Streptococcus, here the cells divide repeatedly in one plant to form chain of cells. Examples of Streptococcus is Streptococcus bicogens and Tetrococcus. These consist of 4 round cells which divide in 2 planes at the right angles to one another. Example is Gaffkia detragena. The next type of bacteria is Sarcina. Sarcina is in this case the cells divided in 3 planes, but they form a cube-like configuration consisting of 8 or 16 cells, but they have a regular shape. Example is Sarcina lutea. In Bacillus, these are rod-shaped cylindrical bacteria which either remain singly or in pair. Example, Bacillus ceres. In Vibrio, the Vibrio are the curved, comma-shaped bacteria and represented by a single genus. Examples of Vibrios, Vibrio cholerae. In Spirilla, this type of bacteria are spiral or spring-like with multiple curvature and terminal flagella. Examples of Spirilla are Spirillum voltans. Thank you. Thank you for watching my video.